Hi everyone, my name is Julia DeFeo and my Biology Day presentation is entitled Effects of Prescribed and Wildland Fire on Arthropod Functional Trait Diversity in Pinelands Ecosystems. I'm working on this project with Dr. John Dighton, Dr. Angelica Gonzalez, and Dr. Michael Gallagher. As many of us know, incidences of wildland fire are becoming increasingly common and severe across the world due to climate change. The use of prescribed fire as a tool to decrease the likelihood of larger fire events occurring is also increasing, and for this reason it is important to understand the effects of fire disturbance on soil arthropod communities, because of the integral role they play in soil health and community function. This is why I have chosen to study the effects of fire disturbance on arthropod community structure. Disturbance regimes can vary, for example from a low frequency to a high frequency of disturbance. And as we move, for example, from a low frequency of disturbance to a moderate frequency to a high frequency, we expect to see a bell-shaped curve where diversity peaks at the intermediate disturbance frequency. We are often most familiar with taxonomy and phylogenetics as measures of community diversity. However, functional diversity is also another fundamental dimension of diversity. Functional traits are phenotypic traits that can affect organismal fitness through their ability to impact organismal characteristics like reproductive ability and nutrient acquisition. In plants, specific leaf area and leaf construction materials are commonly studied, but in arthropods, and in this study, we are investigating trophic guild, body size, and dispersal mode. Trophic guild provides information about competition and food sources. Body size informs us of resource acquisition within the community, and dispersal mode provides information that helps us to investigate colonization dynamics. There is strong support for the intermediate disturbance hypothesis as a predictor of taxonomic and functional diversity in plant communities. In this study, which looked at plant richness along a gradient of increasing anthropogenic disturbance, both taxonomic and functional diversity were found to peak at intermediate disturbance severity. As you can see in each of these graphs, both species and functional richness peak at intermediate disturbance intensity. Therefore, we can ask the following question: how does fire disturbance affect taxonomic and functional diversity of soil arthropods? Based off this background information, we hypothesize that moderate disturbance will favor high taxonomic and functional diversity, in accordance with the intermediate disturbance hypothesis. And furthermore, we expect that species functional traits such as body size, dispersal mode, and trophic group will be significant drivers for arthropod species composition post-fire. Specifically, we expect that moderate fire severity will correspond to peak taxonomic and functional diversity, and that the organisms present in high fire severity areas will have high colonization ability, and conversely, the organisms present in low fire severity areas will have high competitive ability. This study is being conducted in the New Jersey Pinelands using 15 independent plots that constitute a gradient of fire severity, and field sampling and plot characterization took place over the summer, using pitfall traps as a sampling method to collect the soil-dwelling arthropods. Back at the lab, in addition to performing soil analyses, the arthropods that were collected over the summer have been identified to morphospecies, and this information will help us determine the taxonomic and functional diversity of the study organisms. Our preliminary results indicate that changes in species abundance are the driver of fluctuations in taxonomic diversity in our study system. In accordance with our hypotheses, species abundance is greatest at intermediate disturbance, peaking in the plots that experienced moderate fire severity and low severity at annual frequency. This work is still ongoing, and by the end of the year we hope to complete the morphospecies identification of our microarthropods, complete the functional trait analysis and determine dispersal mode, trophic guild, and body size for each organism group, and we also hope to conduct statistical analyses. In conclusion, I would like to acknowledge and thank my funding sources for their support of this work, my advisors, Dr. John Dighton, Dr. Angelica Gonzalez, and Dr. Michael Gallagher for their support and assistance in facilitating this project, and Alexis Everland, Steve Schultz for their assistance in the lab and the field, and the New Jersey Forest Fire Service for their assistance in selecting and gaining access to field sites. If you have questions about this work or would like to learn more, I hope that you'll join me during Biology Day for my presentation on this project. Thank you.